Hello student, I am Dr.B.K.Barun from Medico Patna, and today class is enzyme. So enzyme is a biocatalyst, and all enzymes are protein, but all proteins are not enzyme, and except ribozyme and ribonuclease P. These ribozyme and ribonuclease P is actually nucleic acid. So these are the nucleic acid, and enzymes actually divided into 6 group. इसको आप याद रख सकते हैं. OT में है लिली. O stand for oxidoreductase, T stand for transferase, H stand for hydrolase, L lyase, I isomerase or Li ligase. And the example is, these are class or subclass, subclass, and main activity of enzyme. So these are divided into total 4 type of classification, such as 1, 1, 1, 1, 1, or 2, 1, 2, 3. So these are the class, subclass, subclass, and then main activity. So oxidoreductase example is dehydrogenase, oxidase, reductase, peroxidase, and the transferase is the hexokinase, glucohexokinase, and hydrolase means there is a hydrolysis. So all digestive enzymes is a hydrolase, and sucrase, invertase, alpha amylase, beta amylase, maltase, lactase, all hydrolytic enzyme, and that is the suicidal lysozyme. All lysozymal activity is also a hydrolase, and without hydrolysis, that is lyase, such as carbonic anhydrase, aldolase, decarboxylase, and isomerase means isomerization reaction, such as glucofructoisomerase, cis trans retinyl isomerase, and that is the ligase. Ligase example is PP carboxylase, RUBP carboxylase, and DNA ligase. So these are the 6 classes. And the factor affecting enzyme is the substrate concentration, Km value, temperature, pH. And this is temperature and pH optima. And what is Km value and substrate concentration? Km value is the Michaelis-Menten constant. And this Michaelis-Menten constant arises from the name of scientist, and it is the Km value is the equal to substrate concentration at which velocity is half of maximum velocity. So this is the definition of Km. And the enzyme activity is maximum at the temperature, that is the around 25-30 degree, and above 40 degree the enzyme is denatured, and 80 to 90 degree some bacteria survive but enzyme denatured, and pH is around 7. So the maximum activity of enzyme at temperature and pH is fixed. This is temperature optima and pH optima. And factor affecting enzyme is substrate concentration, Km, temperature, and pH. And enzyme is highly specific. This is the property of enzyme. And enzyme is always protein, but all proteins are not enzyme. And the cofactor is the prosthetic group, coenzyme, and metals. That is 3 cofactors. If hemoglobin, that is prosthetic group in heme, it is tightly attached with the protein part of enzyme. Enzyme has usually 2 parts. One is protein part, another is non-protein part. Protein part is apoenzyme, non-protein part is prosthetic group, that is cofactor, coenzyme, and metal science. So the prosthetic group is tightly bonded with the apoenzyme.